بسم الله الرحمن الرحيم والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا ونبينا محمد وعلى آله وصحبه أجمعين. Now this is the first lecture in the cardiovascular system microbiology department and it is about myocarditis and pericarditis. And these are the topics that we are going to discuss during the whole block. And these are the objectives of our first lecture. Myocarditis means inflammation of the heart muscle, and it is characterized by some immune cells infiltration within this heart muscle. And this inflammation can result in myocardium destruction, and sometimes it can extend up to the valves of the heart. But all of this should happen sparing the coronary artery, because once coronary arteries are obstructed, the disease name will be changed from myocarditis to myocardial infarction. Myocarditis can sometimes be so severe and can lead to necrosis of the myocardial tissues. A lot of myocarditis cases can end or complicated by a condition known as dilated cardiomyopathy. Dilated cardiomyopathy means that the heart size is enlarged, but it's not working efficiently. So although the heart looks larger than normal, but the function is impaired. Here to the left, you can see an autopsy from a normal heart, and a heart of a patient died because of dilated cardiomyopathy. Now you can see that the heart size is enlarged than normal, and the left ventricle size is much enlarged. Look at the cavity of the left ventricle, it is enlarged, and you can see the white tissues which represent fibrosis. But although the left ventricle cavity is enlarged, the thickness of the heart muscle is decreased, and that's why this heart is not working efficiently and is not pumping the blood promptly. These patients might end having heart failure, arrhythmias, and they might die because of these conditions. You can see in this slide the normal histology of the heart muscle. You can see the cardiac myofibril, the intercalated disc, and the nuclei. This histology is completely distorted during the cardiomyopathy. Look at this picture. Here you can see the necrotic heart muscles and the infiltration by immune cells. Mostly in this case, these are lymphocytes due to viral infection. Myocarditis can happen either due to infectious causes or non-infectious causes. Infectious causes include viruses, bacteria, protozoa, helminths, and fungi. So plenty of organisms can cause it, while the non-infectious causes can happen due to exposure to toxins, drugs, or due to autoimmune reactions. Myocarditis can happen due to non-infectious causes, just like the autoimmune reactions. Some of these autoimmune reactions happens after exposure to an exogenous antigen, just like the case of acute rheumatic fever, which happens after the infection by the bacteria Streptococcus pyogenes. Or it can happen sometimes against an endogenous antigen, just like the case of the systemic lupus erythematosus and systemic vasculitis, where the immune system is attacking now some self-antigens. In both of the cases, myocarditis is going to start. Other non-infectious causes include cardiotoxins, and these toxins can affect the myocardium either by initiating a hypersensitivity reaction, just like the case of toxic shock syndrome toxins produced by Staphylococcus aureus and Streptococcus pyogenes. These toxins work as super antigen and they can lead to a huge inflammation that can affect the myocardium. Snake venoms and some antibiotics can also initiate hypersensitivity reactions with severe inflammation that can destruct the myocardium. On the other hand, The carbon monoxide, the heavy metals, and ethanol, they have a direct lethal toxic effect on the myocardium. So they by themselves have their toxicity on the myocardium. Now, the first question for this lecture is that you have to mention one bacterial toxin that have a direct toxicity on the myocardium. Rheumatic fever is a well-known complication that happens after the infection by the group A streptococci, which is Streptococcus pyogenes. The patient in this state suffers of acute symptoms of rheumatic fever. And then with the recurrent infection by the bacteria and the ongoing inflammation, he's going to end up having rheumatic heart disease. Now you have to answer these three questions regarding this topic. First, you have to tell us about the virulence factors of group A streptococci, and then which one of these virulence factors is leading to establishment of this condition, the rheumatic heart disease. Then you have to tell us by which mechanism had the immune system lost the tolerance against the heart valve and why it is now recognizing the heart valve as non-self and attacking it. Lastly, you have to tell us about the immune mechanism that had led to this tissue destruction. Symptoms of acute rheumatic fever appears usually 1 to 4 weeks after infection by group A streptococci. Majority of patients present with symptoms of pericarditis, which can involve the myocardium, the endocardium, and the pericardium. Other symptoms can happen in the form of migratory polyarthritis, and they are called migratory because the pain is spread from one joint to another or from one group of joint to other groups. And while some joints start to improve, others start to become more painful for the patients. Some patients might also present with subcutaneous nodules, which are completely painless nodules found near the joints. Skin manifestations appear in the form of erythema marginatum. It is called erythema because the rash is red in color, and it's called marginatum because the skin rash has well-demarcated margins. Some patients might present with involuntary movement called chorea, and this means that some neurons are also affected during this disease. Rheumatic heart disease happens due to recurrent infection by group A streptococci, and the ongoing inflammation, which is now becoming chronic, usually affects the mitral valve and the aortic valve. Affection of mitral and aortic valve and sometimes tricuspid valve is going to predispose these patients to bacterial endocarditis and might also result in heart failure and death of these patients. Now we'll talk about the infectious causes of myocarditis, starting by the protozoal infections. The first protozoa we're going to talk about is the causative agent of Chagas disease. Now, worldwide, viruses are the most common causative agents of myocarditis, except in Central America and South America, where the protozoa Trypanosoma cruzi is the most common causative agent of myocarditis that appears in the form of Chagas disease. This protozoa has many transmission modes, but the dominant one is being transmitted through the winged bug called Triatoma, and sometimes called the kissing bug because it tends to bite the patients in their faces. But this Triatoma doesn't introduce the protozoa during biting. The protozoa is actually present in the feces of this bug. So after biting, the patient is going to scratch the bite area and rub the feces of the bug, which contains the Trypanosoma cruzi, to inside the wounds, and in this way it's going to enter inside the body. Or sometimes it can bite near the mouth or near the eyes or near the nose, and through the mucous membrane, the Trypanosoma cruzi can enter to inside the body. Other modes of transmission also include vertical transmission when the infected patient is a pregnant lady, or by blood transfusion or by tissue transplantation. The life cycle of Trypanosoma cruzi includes 3 stages. The first one, the metacyclic tribomastigote, is the infected stage. That means it is the stage that is contained within the feces of the triatoma. The other two stages are the tribomastigote, which is the flagellated form, which is present in the blood. Now, mastigote means flagella. The other form, the third form, is called the amastigote, which means it doesn't have a flagella, and this form is found in the tissues. So if we collected a blood specimen from the patient, we will find the tribomastigote stage. If we collected a biopsy, then we will find the amastigote stage, while the metacyclic tribomastigote is found only in the feces of the triatoma bug. Which stage of the parasite produces the localized infection inside the myofibrils of the heart? And what are the cardiac abnormalities induced by this infection? You can see here the triatoma bug. Triatoma sucks its meal of blood from the patient, and once its stomach is engorged by the blood meal, it's going to defecate on the skin. So you can see here the feces of the bug, and this feces contains the metacyclic tribomastigote. Now, usually the bite is painful, so the patient is going to scratch the area, and this is going to rub the feces with the metacyclic tribomastigote to inside the skin. Once inside the body, the metacyclic form is going to transform into the amastigote and enters inside the tissues and start to multiply there. Once the tissues ruptures, it's going to release the tribomastigote form. Tribomastigote form can either enter again inside the tissues and transform into amastigote and start to multiply and increase in number, or it can go to the bloodstream and wait in the blood vessels for the triatoma to come and suck it again and then infect other human being. Chagas disease is also a zoonotic disease. It can affect many mammals. If these mammals are affected, triatoma can also transmit the infection between them and to the human also. So again, you can see here the triatoma bug is sucking the tribomastigote with the blood meal from this patient and releasing its feces that contains the metacyclic tribomastigote. Metacyclic tribomastigote is going to enter inside the tissues and lose the flagella and become a mastigote, which is able to multiply by binary fission. Now, once the tissues are ruptured, tribomastigote is released. Some tribomastigote are going to enter again inside the cells, multiply there and in the form of amastigote, or some of them might wait in the blood vessels for the next triatoma to come and suck them and transmit them to other patients. Here you can see a blood film stained by Giemsa stain, and you can see the tribomastigote with its flagella. This is a biopsy from a muscle, and it contains the amastigote form, numerous amastigote forms intracellular, multiplying and increasing in number. And that means infected patients by Trypanosoma cruzi are losing tissues, losing mostly muscles because of the multiplication of the Trypanosoma inside their tissues. Chagas disease is classified clinically into two stages, the acute stage and the chronic stage. Acute stage presented first by presence of localized swelling at the site of the entry of a protozoa called chagoma. If the chagoma happens in the face, it's called Romanas sign. And then the patient is going to suffer also of some vague mild symptoms, just like fever, headache, diarrhea, or vomiting, and his liver and his spleen might also be enlarged. Now, this is chagoma, which is a localized swelling in the skin at the site of the entry of the metacyclic form. If chagoma happens in the face, it is called Romanas sign, which is swelling of the eyelid, either because the patient has accidentally rubbed the feces that contain the protozoa to inside his eyes, or because the triatoma bug had bitten him near the eye. As I told you before, triatoma is sometimes called kissing bug because in most of the patients it tends to bite them in their faces. If the patient didn't receive appropriate treatment during the acute stage of Chagas disease, they're going to present 10 to 20 years later in the chronic stage of the disease. Now, during this period, the mastocyte of Trypanosoma cruzi is multiplying inside the tissues, many systems of the body, including the myocardium. The multiplication of the mastocyte inside these tissues lead to tissue destruction and loss of these tissues. And now the immune system is responding by a cell-mediated immune response. So we're going to have T-cell delayed hypersensitivity reaction, which is going also to destruct the tissues that contains the mastocyte. And then autoantibodies are also produced against the actin, the myosin, and the laminin. So now the antibodies and the immune response are destructing more tissues. So three mechanisms collectively are acting against the myocardium: the multiplication of the protozoa, the immune reaction against the protozoa, and the autoimmune reaction against the myocardium, even those which are not affected. So now myocarditis had started, and it will end result by dilated cardiomyopathy, which can lead to cardiac arrhythmias, heart failure, and death. Other systems in the body can also get infected, and this include the GI tract. So the patient might present with a megacolon or megaesophagus, and sometimes they might present with megaureter. And the parasite can also multiply inside the neurons and lead to central nervous system manifestations. You can see in this slide the autopsy of a heart of a patient died because of Chagas disease, because of dilated cardiomyopathy. Look at the size of the heart. It is highly increased. Look at the chamber size, highly increased, and you can see also the white fibrotic tissues, and you can see that the muscles of the heart are decreased. So the ratio of the thickness of the muscle to the cavity of the ventricle had been decreased, and that's why the heart did not function well. Look at this X-ray. You can clearly see that the heart size is enlarged than normal. Here you can see an intestine during a surgery to restrict this part of intestine. This is the megacolon. The size of the colon is much more large than normal, and it is completely paralyzed. That's why they have to be resected and taken out, because they can lead to many complications, including septicemia. We are still talking about infectious myocarditis, still talking about the protozoal causative agent of myocarditis, and this time the protozoal name is Toxoplasma gondii, the causative agent of toxoplasmosis disease. Toxoplasmosis is usually a mild self-limiting disease, unless the patient who had acquired the protozoa was immunocompromised, or he was a fetus inside the uterus when his mother contracted the protozoa for the first time in her life. So those who are at risk are the fetuses inside the uterus and the immunocompromised patients. Pregnant ladies themselves are not at risk of having severe disease. It is the fetus inside the uterus who might suffer of congenital anomaly. Toxoplasma gondii had many stages in its life cycle, and it has also many transmission modes. Among these transmission modes, it can be transmitted by ingestion of the oocyst from the cat feces, or by ingestion of the tissue cyst in undercooked meat, or by blood transfusion or transplacental from the mother to the fetus inside the uterus. These are the transmission modes of Toxoplasma gondii. Now, cats release the oocyst stage with their feces. If the oocyst stage was ingested accidentally by the human with food and water, they can cause the infection, or they can coat it if they ingested undercooked meat that contain the tissue cyst in some animals. Transmission can also happen by blood transfusion or by tissue transplantation, and can happen also by vertical transmission from the mother to the fetus inside the uterus. If the fecal oocyst was ingested by some small mammals, it can infect them and turn into tissue cyst inside their organs, And if the cat feeds on these infected animals, it can cause the infection herself and now start to release the fecal oocyst, which can again cause the infection to other mammals, including the human. Viruses are the most common causative agents of myocarditis worldwide, including the Coxsackie viruses group B. Coxsackie viruses belong to the family Picornaviridae, which means small viruses, and they belong to the genus Enteroviruses. Coxsackie viruses are non-enveloped, single-stranded RNA viruses, and there are two groups of them, Group A and Group B. Why are the Coxsackie viruses classified as enteroviruses? Group A and Group B Coxsackie viruses, both of them can cause upper respiratory tract infections and they can cause also aseptic meningitis. But Group A usually tend to infect the skin and the mucous membranes and can cause diseases such as herb angina, which means the patient is having blisters and vesicles in his mouth, usually in his soft and hard palate, which make it difficult to swallow because of the pain. Other disease caused by Group A Group A viruses, Coxsackie viruses, is the hemorrhagic conjunctivitis. You can see here that there is hemorrhage beneath the conjunctiva of the eye. It can also cause a disease in the childhood called hand, foot, and mouth disease, where the patient has painful blisters in the palms of the hands, the soles of the foot, and surrounding his oral mucosa. All of these diseases are mild and they are completely self-limiting. On the other hand, Group B Coxsackie viruses tend to infect the connective tissues, including the myocardium and the pericardium. What are the clinical presentations associated with a respiratory and cardiac infection by this virus? Group B Coxsackie viruses invade the pharynx or the gut wall and then hematogenously spread to other tissues, just like the muscles, the myocardium, and the pericardium, and an inflammation is going to be evoked there. This inflammation, of course, contains lymphocytes because these are viruses. Several mechanisms are going to lead to myocardium destruction during the infection by Group B Coxsackie viruses. First of all, of course, the presence of the viruses themselves inside the cells of the myocardium and their multiplication there. And then we have the immune response against the infected tissues in the form of natural killer cell, cytotoxic T lymphocytes, T helper cells, and B lymphocytes. And in this case also we have autoimmune destruction of the myocardium because some antibodies are produced against some self-antigens, just like against the myocyte ion transporter and against the signal transduction proteins. Because the inflammation now happening in the form of autoimmunity, it's going to become a chronic inflammation leading to chronic myocarditis and then dilated cardiomyopathy and then heart failure and death of the patients. Among the viral causative agents of myocarditis, we have also the newly emerged disease, the COVID-19 disease caused by SARS-CoV-2. SARS-CoV-2 belongs to the family Coronaviridae, which are RNA viruses that always carries an RNA-dependent RNA polymerase enzyme to make its own messenger RNA. This virus enters to the cells that have the receptor, which is the angiotensin converting enzyme number 2, which is present in multiple tissues in the body containing the myocardium. During COVID-19 infection, the myocardial effect can appear either in the form of myocardial injury that can be detected by elevated troponin, and this is very common, or it can happen in the myocardial injury accompanied by shock and arrhythmias in a quarter of the patients, or rarely the patient might present with only arrhythmias. Now our question is, what mechanism you think had led to myocarditis during COVID infection? And what is the type of COVID vaccine that is reported to cause myocarditis? This is the life cycle of the SARS-CoV-2 virus inside the myocardium and the inflammatory response against the virus. Among the viruses that can cause myocarditis, we have also the virus called parvovirus B19, or sometimes called erythrovirus B19. This virus belongs to the family Parvoviridae, which means also small or tiny viruses. Parvovirus B19 is an non-enveloped icosahedral DNA virus. Very few DNA viruses are non-enveloped, so I think that you have to remember this. The parvovirus B19 has a main target in our body, which is the immature RBCs. So red blood cells precursors in the bone marrow are the major target for this virus. It can be transmitted by many modes. The most important mode is by inhalation of respiratory droplets. It can also be transmitted by blood transfusion or vertically during pregnancy from the pregnant lady to the fetus inside the uterus. Parvovirus B19 can cause many clinical diseases depending on the age of the patient, his gender, and his clinical situation. These diseases include the fifth disease, autoimmune arthritis, aplastic critis, congenital disease, and myocarditis. In the case of congenital disease, the baby is suffering of severe anemia inside the uterus because the virus is multiplying inside his bone marrow and is destructing his RBCs. He might end with heart failure and death intrauterine. So in a lot of cases, these babies might require emergency blood transfusion intrauterine inside the uterus to save their lives. Fifth disease is a very common childhood disease. It is called fifth disease because it is one of 6 diseases that manifest with mild fever and skin rash. It has another name, which is erythema infectiosum, because of the color of the rash and because it's highly infectious. It is contagious because these babies or these infected children are generating the viruses in the form of droplet, which can be inhaled by other children and have the same symptoms and signs. The disease can also be called slapped cheek disease because the rash is mostly concentrated in the cheeks of the children as if someone had slapped them. Our question is, what is the cause of skin rash formation in the case of fifth disease? By which mechanism is the rash formed? So after acquiring the virus by 4 to 14 days, the symptoms will start by a mild fever, runny nose, headache, and then a concentrated rash in the face is going to appear and sometimes spread to other body parts. These children are affected by parvovirus B19. They are suffering of fifth disease. Look at the rash concentrated in their cheeks. That's why it is called slapped cheek disease. Look at this baby, this child posing for the camera, very happy and smiling, and they tell you that this disease is mild and self-limiting. Autoimmune arthritis can also happen as a complication of parvovirus B19 infection. And usually this affects the young ladies. It happens due to formation of immune complex, antigen antibody complexes that deposit inside the joint. Infection by parvovirus B19 can cause an emergency situation, a fatal disease called transient aplastic crisis. And this happens only in those who have sickle cell anemia, thalassemia, or spherocytosis. And our question is, why do these patients suffer of aplastic crisis? Bacteria don't usually cause myocarditis unless the patient has immunocompromission or as a complication of bacterial endocarditis. If the patient has normal immunity, then very few bacteria that belong to either spirochetes or the rickettsiae can cause myocarditis in his situation. Among the spirochetes, there is Borrelia burgdorferi, or the Treponema, the causative agent of syphilis, or the Leptospira. And among the rickettsiae, the causative agents of Rocky Mountain spotted fever can cause also myocarditis. Borrelia burgdorferi is the causative agent of Lyme disease, is a gram-negative spirochete. Lyme disease is a zoonotic disease that is transmitted by tick bite, ticks of the genus Ixodes. Lyme disease happens in 3 stages. The first stage manifests in the form of a skin rash called erythema migrans. If the patient didn't receive treatment, he's going to go into the second stage when Borrelia hematogenously spread to other body parts, including the myocardium, causing myocarditis and cardiomyopathy, and can also cause meningitis and neuropathies. And if this stage also didn't get treatment, he's going to go to the chronic stage with chronic skin infection, central nervous system infection, joints abnormalities. Our question is, we want you to name other co-infections that can happen with Lyme disease, and why are those co-infections happening? Why do these diseases can happen sometimes together? Borrelia are gram-negative, spiral-shaped bacteria, but they usually stains weakly by the gram stain. And here you can see the ticks, the exodus gene that can carry the Borrelia bacteria. And this is the rash caused by or caused during the Lyme disease, which is called erythema migrans. And Lyme disease is a zoonotic disease transmitted from usually deers or from small mammals to human. Cardiovascular syphilis is a disease caused by the spirochete Treponema pallidum, which is transmitted sexually, transplacentally, which is vertically, or by blood transfusion. Myocarditis usually happen in the latest stage of syphilis, the last stage of the disease, the tertiary stage. It happens in 10% of the cases. This stage usually happens late after the infection by about 10 to 30 years. And it can appear in the form of aortic lesions, just like aortitis or aneurysm, or it can happen due to destruction of the myocardium, either by autoimmune antibodies, just like the anti-cardiolipin antibodies, or by cell-mediated hypersensitivity reactions against the myocardium. Diagnosis of syphilis is usually attained serologically by one of these tests, because, as you know, the treponema cannot be stained by the gram stain, and it cannot be cultivated on ordinary media. So syphilis can be either diagnosed serologically, or it can be diagnosed using special types of microscope, just like the dark field microscopy, where you can see the bacteria still motile, or by using the fluorescent microscope. You can see here the spiral-shaped treponema. Bacteria that can also cause myocarditis, the Brucella, Corynebacterium, Neisseria, Haemophilus, and Actinomyces. These bacteria transmit to the myocardium hematogenously, while the Staph aureus and the Streptococcus pyogenes and other species of Streptococci, they spread to the myocardium from the endocardium. So they cause endocarditis first, and then the infection is spread to the myocardium. Some fungi, including Aspergillus, can also cause myocarditis. Myocarditis by Aspergillus happens only in the form of invasive aspergillosis on those who are immunocompromised and neutropenic patients, where the virus has the ability to spread from the lung to other tissues and invade them. Some helminth, including Trichinella spiralis, can also cause or can also infest the myocardium and cause myocarditis. Trichinella spiralis is a nematode that causes a disease called trichinosis. It is transmitted by ingestion of undercooked meat, usually pork meat or wild animals meat, that contains the encysted larvae, which is considered the infected stage. So someone is going to ingest the undercooked meat that contains the larvae, and inside his intestine, this larvae is going to come out of the cyst and become adults, males and females, and they mate, and then the female starts to lay larvae inside the intestine. These very small larvae are hyperactive, and they can be carried by the bloodstream to other body parts. Usually they prefer to stay in some muscles. All muscles can be infested by these larvae. Once the larvae had reached the muscles, they are now large in size, and they can no more travel through the capillaries and go to other tissues. So they are trapped inside the muscles, and then they will make a cyst around themselves and lie inside the muscles in a spiral form. And that's why they are called Trichinella spiralis, because they coil around themselves and create a cyst around themselves and then stay latent inside the muscles of the patients, including the myocardial muscles. The diagnosis of myocarditis is not an easy task. It's going to depend on the clinical picture of the patient, and they're going to do some cardiac examination and cardiac investigations, just like the ECG and the troponin level. If myocarditis was caused by an infectious agent, then the C-reactive protein and the ESR are expected to be higher than normal. You can rely on serology by finding the IgM against certain non-causative agents of myocarditis for diagnosis. In some patients, the diagnosis can rely only upon taking a biopsy from the myocardium, and then you can see the necrotic tissues, distorted myofibrils, and the infiltrating lymphocyte to diagnose the case. Pericarditis means inflammation of the pericardium of the heart, the chest that encloses the heart. Just like the myocarditis, it can be either caused by infectious or non-infectious agents. Infectious agents can manifest in acute disease or chronic disease. If it is acute pericarditis, then think first of the viruses. Among the viruses, the most common causative agents are again the Coxsackie virus B. Bacteria can rarely cause pericarditis, and if it does, the mortality rate is very high. Bacteria can reach the pericardium either hematogenously during bacteremia or by spreading from the lung and through the pleura during pneumonia to the pericardium. Most common causative agents are the Haemophilus influenzae and Streptococcus pneumoniae, which are the most common causative agents of pneumonia, or it can happen due to infection by Neisseria meningitis or by Staphylococcus aureus or Streptococci group A. Chronic pericarditis happens only in those who are immunocompromised, just like the HIV patients if they are infected by the mycobacteria, so tuberculous pericarditis. Or it can happen due to infection by certain fungi, endemic fungi in certain parts of the earth, and these just like the histoplasma and the coccidiosis. By this we had finished our lecture. Thank you for listening.